today. I apologise for the technical difficulties, but hopefully we'll have a memorable for the rest of the afternoon. So, welcome to this public lecture about findings of our crowdfunding.com funded seaweed project. So, would you like to see me with that? So, there are a lot of problems on a global scale. Poverty and malnutrition are huge problems in the developing nations, whereas in developed nations we have problems with obesity, heart disease and diabetes. We've got massive population increase. We've projected our global population to rise to 9.6 billion by 2050. Along with this, we have climate change issues, a warming climate, and the array of effects that are affecting both our terrestrial and marine environments associated with that. Our water resources are highly limited. Although we're a wet planet, only 3% of our global water sources are fresh water. And these are in high demand, and particularly high demand with an increasing population. Disease is on the increase and potentially an increasing problem under climate change scenarios as well. And so along with this, we have increasing issues about food security. So what's any of this got to do with seaweeds, you might be asking? Well, we think that seaweeds are potentially part of the solution. For a start, seaweeds don't require our precious freshwater reserves to grow. They grow in the ocean, in the salt water, so we don't need to water our seaweed crops, unlike our terrestrial crops and terrestrial farm systems. Seaweeds use nutrients and draw down carbon dioxide, both of which are increasing problems in our aquatic environments with increasing eutrophication from urban and agricultural runoff and increasing CO2 through fossil fuel burning and atmospheric CO2 dissolving into the ocean. Obviously, seaweeds don't require the use of land, so as population increase continues and land becomes even yet a limiting resource and the battle between land for agriculture and land for people continues, we can potentially look to our oceans to farm some of the food that we require for sustaining that increasing population. The other thing about seaweeds is they've got incredibly fast growth rates. So some species can grow up to 6 centimetres per day. So you can almost see them grow before your eyes. So this has got incredible potential for rapid turnover of biomass and rapid food production. Now, the other thing that's great about seaweeds for feeding the poor and feeding the world, indeed, is that they're nutritionally rich. So they're good sources of protein, essential fatty acids, which I'll talk more about today, and fibre and a range of other health benefits. And so a range of other compounds that have got demonstrated health benefits. So, and there's a great big list of anti-things that are good for us that seaweeds provide. So they've been shown to reduce inflammation, provide antioxidant capabilities, antiviral, antibacterial, anti-cancer. Regular consumption of seaweeds has been shown to reduce obesity and diabetes and heart disease associated with those problems, and also protect the brain against Alzheimer's disease. So there's a whole range of potential health benefits that can come from regular consumption of seaweeds. But just because something's good for us doesn't mean we're going to eat it. And so when I ask my first year students, these are the sorts of response I get, and these are the sorts of images that most people think about in Australia. Seaweed is the smelly, slimy, stinky, gross, yucky stuff that's washed up on our beaches. And it doesn't matter whether it's a cloudy, stormy beach in Warrnambool in winter or a nice sunny beach in France in the middle of summer. It's still stinky, gross, smelly yuck, and why would you want to eat that? Well, we think there's lots of reasons you should be wanting to eat that. Maybe not the stinky, smelly stuff that's washed up on the beach, but certainly the nice fresh stuff picked from our beautiful clean Australian oceans. So large-scale commercial production of seaweeds began in Japan in the 1960s, and it's now estimated that about 10,000 tonnes of seaweed product are harvested globally. This amounts to an annual value of about $6 billion US dollars, the large proportion of which is for human consumption. So about $5 billion of that $6 billion product is for human consumption. So this is the main use of seaweeds across the world. And there are a range of other uses such as food additives and additional additives for gelling compounds in toothpaste, beer, paint, all sorts of things, and also pharmaceutical and cosmetic industries. But our focus is on looking at seaweeds as food, and this is the greatest market, as I said. So why are we interested in seaweeds in Australia, and why are we interested in it now? Well, basically sitting here in the Warrnambool campus of Deakin University, we are in a globally significant biodiversity hotspot for seaweeds. Southern Australia has the greatest biodiversity of seaweeds of anywhere in the world, and any regional area in the world, and very high levels of endemism as well. So about 62% of the species that we find on our southern Australian shores are not found anywhere else in the world. So we sort of figure on a matter of probability, there's got to be something good out there to eat. We just haven't looked for it. But the other question is, you know, assuming there is something good out there to eat, can we actually get people to eat it? So this project was developed to try and, in the long-term goal, assess the potential for a viable and sustainable seaweed industry based in southeastern Australia. And in the short term, what we were trying to achieve with the research that I'm going to present to you today is to assess the palatability of a select group of Australian seaweeds, and also to compare the nutritional value of these seaweeds to others that are already commercially available on the market. So first we had to get the funding, and we did this through the Possible.com partnership with Deakin University and Research My World. And so we had a whole lot of media interest, a whole lot of public interest, and a whole lot of people putting in small to large amounts of money that enabled us to fund this research, which was absolutely fantastic, and I thank you all out there. We then, of course, needed some guinea pigs to taste our seaweed, and we also put out a call into the local area to recruit volunteers to taste our seaweed. But we wanted to recruit volunteers that had some experience with eating seaweed. When the local, or the general Australian reaction to seaweed is smelly, stucky, stinky stuff, then we want to make sure that we're not feeding this to people that just generally have a dislike for seaweed, so they score all our seaweeds as bad just because all seaweed is bad. We wanted to have a group of tasters with a discerning audience, and we've got many of those here today with us. So again, thank you all. We then had to set about looking for which species that we were going to choose to target, and with the plethora of species that we had potentially available, we narrowed it down by firstly looking at species that we thought are likely to be good to eat. And I spent a lot of time, about 3 years of my life living in Japan, and so eating seaweeds became a common thing for me. So I had a relatively good background knowledge about the sorts of species that were eaten, at least in Japanese diet, and the ways they were prepared. So I could look at our Australian flora with a new set of eyes and think about which species did I think were likely to be good to eat based on the phylogenetic relationships, so the relationships with species that were already commercially on the market in the Northern Hemisphere, and also the similarity of structural form and texture. So we used these to guide us in our seaweed selection for these initial trials. We also looked at species that we thought were likely to have potentially high nutritional value. We thought we may as well start at the top end of the market, and again, look for ones that we think are going to be most likely to be both palatable as well as nutritional. And importantly, we looked for species that were common. So we figured in the first instance, if we're going to develop the potential for a seaweed industry in Australia, it's likely to be based, at least in the first instance, on wild harvest before it can go to commercial agriculture. So for that reason, we need to be targeting in the first instance species that are abundant, because we need to be able to set up a sustainable wild harvest if that's the case. And we then pitched these species against a group of commonly consumed Japanese species. So on the the right-hand side of the slide, you can see the list of species that we've got there. So we've got one red algal species, one green, and a range of brown algal species, all a range of different sort of kelp species. And we compared that to the Japanese nori, which is a red alga, that's the species that's wrapped around the sushi, and 3 different brown algae species that are common in the Japanese: hijiki, kombu, and wakame. And so this is what our Australian species look like. So those names may mean nothing to you, but maybe for those of you that spend a bit of time out in the ocean, these pictures might ring true up. So certainly these, the range of brown algal species that we've got are common and abundant on our southern Australian shores, and also occur in groups that are relatively diverse. Okay, so we then went about having to collect these species, and we're interested in looking at the temporal variation in the nutritional value as well, because we know from other research in other areas that there are differences in the nutritional composition of seaweeds in different times of the year and also different locations. So with the limited amount of funding that we had, we thought it was interesting to do a short temporal study to compare the nutritional value across autumn and winter. And importantly, we were collecting from clean local sites. Seaweeds do have the potential to accumulate toxins. So when you are collecting them from the ocean, it's really critical that you are collecting them from clean, unpolluted sites. And I'll talk a little bit more about that for some of our future research at the end. So we collected these samples, we blanched them, we dried them, and then when we came to prepare them for our dishes, we rehydrated them and put them up according to the standard practice. And this is relatively standard practice for preservation of seaweeds in Japan. And so we mimic those processing procedures so that we can compare like with like. And we figured it's most likely that the processing itself may have an effect on the nutritional value, so we wanted to make sure that we were measuring the end product rather than the start product. So we then use these seaweeds in a range of taste trials, which I'll go on to explain, and then some preliminary nutritional analyses. So firstly, to look at the taste trials. So when we were looking for inspiration for our dishes, my children thought that we should look to the Octonauts, their favourite cartoon, and the menu to kelp. But I thought perhaps we should look to something with a bit longer history of eating seaweeds, and so instead we chose some Japanese cuisine. And so we based our dishes for our taste trials on these 2 dishes, which hijiki no mimono, which is a simmered seaweed salad. I guess we broadly translate it to in English, based on hijiki seaweed, which is the Japanese seaweed that's commonly used in this dish. And then the miso soup using a seaweed stock, so a seaweed dashi stock, as the basis for a miso soup. But we didn't want to confuse the taster's palate with a range of other ingredients, such as the peanuts and the carrots that you see in the hijiki dish, and the tofu and mushrooms and other things that we put into the miso soup. So we trimmed them down so we had the base dishes, so you could really taste the presence of the seaweeds in those dishes. So for our taste test, we had 3 Australian dishes and 1 Japanese dish for each of these, 3 Australian species and 1 Japanese species for each of the dish styles. And so for the soups, as I said, we stripped out the additional ingredients and just had the basic stock and the addition of the miso. And then for the seaweed salads, we had 2 Sistropha species, a Philotrypia species, and Sargassum which is the hijiki. So the kombu in the soup and the hijiki in the salad, and then 3 Australian species for each dish that we thought were likely to be comparable in properties to those Japanese species. We then asked we presented these dishes to our tasters in blind surveys, and we asked them to score them. Firstly, the first time they saw them, their overall satisfaction with the dish on a 9-point scale from liked it extremely down to disliked it extremely. And then we presented each dish again a second time round and asked them to assess a range of attributes from the general appearance, colour, smell, texture, flavour, sweetness and saltiness. And some of those were assessed before they tasted the dish and some of them were assessed after they tasted the dish. And so, as I said, we did these in blind taste trials, so our tasters didn't know what they were being exposed to, and they were encouraged or encouraged not to talk to each other or discouraged from talking to each other so that we could get independent assessments of the taste and palatability of these dishes by each of our tasters. Now, so I'm just going to present some of these results, and for some of you, you might not be used to seeing data in this way. So this is a multi-dimensional scaling plot, and basically this plots all the different data from our tasters, and it plots it in space, so based on the general attributes. So points that are closer together are more similar than points that are further apart. So if you get a clustering of one particular species together and a tighter clustering of other species apart, means they're quite different to each other. If you get this sort of shotgun approach here, it means there's no difference. And here we've got the Japanese kombu in red points and the Australian species all represented by different symbols in blue. And what you can see here is that there's no clear difference in the suite of attributes that we asked our tasters in and how much they like those different attributes amongst the different species of seaweeds that we use for our soup stock. We can see this again here. We've got the overall satisfaction, and you can see there again there was no significant difference in the overall satisfaction of the different species. So the 3 Australian species are here and the Japanese species in the red here. So they're all scoring quite highly in around 8, a score of 8, which is second from the top in the 9-point scale. And similarly, when we looked at the individual attributes, just looking here at the flavour, there was no difference amongst the species. The only difference we found indeed out of any of the attributes between the different species that we looked at for the soup was this difference in colour. And so we can see this here where the Alecia, one of our Australian species, was scored less favourably for colour. But interestingly, that didn't actually affect the people's opinion of the flavour or the overall satisfaction of the dish. So it doesn't look quite as nice, but it still tastes good. So take a message from that. Now, for the salad, we've got a bit more clumping. So here we have the Japanese species in the red and the Australian species in green symbols. And here we did actually have significant difference in some of the attributes between the Japanese species that are clumping tighter together and the Australian species that are more dispersed. And this was primarily due to differences in texture. So this graph here represents the different attributes that contribute most to the separation of species here in this graph. So this is simple analysis data for those that are doing that data analysis. And we can see that nearly 50% of the difference between species was attributable to differences in texture. And we can see, if we look at that in a different way, we can see again, in this case, there were differences in the overall satisfaction with the different species that we used for the salad dish. And so here we have the Australian species here and the Japanese species on the right-hand side. There's 3 Australian species on the left, the Japanese species on the right. And you can see that, generally speaking, the Japanese species, the hijiki, was scored more favourably than the Australian species. But these two species of Cystophora teleosa and Philotrichia decipiens were scored more favourably than Cystophora polycystina. So, of the Australian species, people are liking these two better than this one, but they're liking the Japanese one better than any of the Australian ones. And that was primarily due to these big differences in texture between the Japanese species and the Australian. And for those of our tasters that are in the audience and can remember those different dishes, there was a distinct difference in the texture of the nigiri compared to the texture of our Australian dishes. So I recently went to China to present the results of this research at the Asia Pacific Biological Forum. And when I presented this research there, one of the Japanese colleagues came up to talk to me afterwards, and he actually said to me that there's a whole lot of processing that goes on with the nigiri before they dry it, that we weren't able to access that information prior to our study. So what they're actually doing is they're blanching it, they're rinsing it and drying it, but then they're actually taking it to the factories and they're boiling it for a number of hours and then drying it again and then taking it to market. So we missed that long boiling stage, and they said they're boiling it for up to 3 hours. So the downside of that, it might improve the texture and palatability of it, it potentially reduces some of the nutritional value associated with that species. So when we look at our nutritional data, some of the differences between the nigiri and the Australian species might be because of this extra boiling and the potential for nutrients to leach out of the nigiri with that long boiling stage. So there's still some work to be done on optimising the processing, but I think these results show that from a palatability perspective, these two species in particular are looking quite promising, and indeed Cystophora tullulosa, from the written comments that we got on the form, Cystophora tullulosa is probably the big hitter in this group of species that we're looking at. And for the soups, they're all looking potentially good. So if we then take a look at what's inside these species and see how they're bearing up on the nutritional perspective. Firstly, we looked at fatty acids. So to explain a little bit about what we're talking about there, we're talking essentially about the good fats. So omega-3 polyunsaturated fatty acids, and so these are fatty acids that people are generally thinking of as fish oils. So EPA, DHA, ALA are terms that general public may well have heard about. And so typically our sources for these are fish and other seafood. So, and relatively recently, krill have been harvested on a large scale for these omega-3 fatty acids, as well as the fish that have been farmed and fish and oil harvested from those for a long time. But they're also present in algae, both microalgae and macroalgae. And indeed, it's only the algal sources that are actually producing these fatty acids. The fish and the seafood, other seafood, are actually getting it from their food source and accumulating them within the body. So the other good fats are the omega-6 polyunsaturated fatty acids. So your LA, linoleic acid. So these are things, the main sources of these that we typically think about in vegetable oils. So your good oils, your avocado, your nuts, your olive oil, for example, and linseed oil. But again, these oils are common and potentially abundant in algae. So in the seaweeds and in the animals that these oils occur in, and indeed in us as well, they have a range of important functions, from energy storage, so storing energy reserves in the oils, but also for structure of cell membranes, delivery of nutrients, and a whole range of metabolic functions. And for human health, they've been shown to have demonstrated their benefits for brain development in particular. So that's been strongly linked to improved infant development with supplementation or high intake of fish oils or omega-3 fatty acids, and also for treatment of individuals with mental health problems. And there are also benefits of eating fish oils for treatment of a range of cancers and cardiovascular disease. So there are good health benefits for trying to get more of these omega-3 and omega-6 fatty acids into our diets. So to do this, we basically performed perform some laboratory analyses to extract the lipids and then separate the fatty acids and identify them based on a range of laboratory techniques that I won't go into full details about today. And what I'm going to talk to you about today is basically just concentrating on these good fats. So we've got a whole suite of fatty acids that we've got, a whole range of data that we can look at in much greater detail and try and work out what they all mean, and we've got different combinations. I'm just going to focus on a few of these key fatty acids that I've talked about so far. So firstly, when we look at omega-3 fatty acids, so in the fish oils, we can see that generally the dotted line here on the ALA graph represents the level at which it's considered to be a good source, a good dietary source of ALA. So above 200 milligrams of ALA per 100 grams of edible product is considered to be a good source of ALA. And we can see that only one of our species tick that box. And on these graphs that I'll show you, the Japanese species are all represented in the orange colours, and the Australian species are represented in blue. So only one of our species ticked that box for the ALA, and that was the codium, which is the green algal species that we looked at. For the EPA, we can see that we've got relatively high levels, with most species somewhere up around 100 milligrams per 100 grams of edible product. But this one here stands out, and this is the nori. Okay, so we've got a huge proportion of EPA in the nori. So this is a real standout compared to all the other species that we tested. But we can see that the Australian species are generally, at least some of them, have got relatively high proportions of EPA there. Now the DHA is relatively low, less than 5 milligrams of DHA per 100 grams of edible product across all the species, and that's fairly consistent with studies for other algae looking at DHA. Generally, they're being reported to be relatively low, and the EPA is a much higher in concentration. But when you combine the two together, this is what is used to assess whether a product is a good dietary source. So a source or a good source, and that's what these two lines represent here. So above 30 milligrams per 100 grams of edible product, you've got a source of these EPA and DHA, so a source of omega-3, and above 60 milligrams is considered to be a good source. And we can see here that a range of our species were considered to be both sources and good sources of these omega-3 fatty acids. But, and again, the standout here is the nori, with this very high concentration of the EPA in particular. So the Australian species are looking pretty good from our fatty acid perspective. And then when we look at our omega-6 fatty acids, we can see that the Australian species are actually outperforming, or at least performing as well as the Japanese species in the orange there. So quite high percentage of omega-6 fatty acids, and generally higher in the Australian species. And this species, Alecia Bellgrove, which is the red algal that we checked, is the standout in this case. So the other type of nutritional analysis that we looked at was proximal analysis, and this looks at moisture content and total protein, lipids, crude fibre, ash, and total carbohydrates. So it gives us an idea of the overall composition of the seaweeds from this gross perspective. Now, if we look firstly at protein, we can see again that quite a lot of our species are having quite high proportions of protein. So this is the percentage protein on the y-axis, and the different species here. We've got the Japanese species in red and the Australian species in blue. And we can see that most of our species have got protein concentrations comparable to that of tofu on a percentage basis. And again, the nori is the standout here. So the Japanese species, the nori, is the standout here with really high percentage protein, with nearly up to, I think, 45% protein. So comparable to beef and soybeans. But of course, that's on a percentage basis, and we know that nori is quite light. You've got to eat a whole lot of nori. So it's similar to the spinach and iron argument with the beef. You know, it's similar on a percentage basis, but you've got to eat a whole lot more product. But clearly it's showing that in your sushi roll, you're getting not just the protein from the fish in the middle of your sushi, but that nori wrapped around your sushi roll is also giving you a protein punch, as well as the omega-3 fatty acid punch. But some of our Australian species are also looking, pretty good with protein values in the range of 5 to 17%. Now, if we look at lipids, we can see generally that overall the total percentage lipids are relatively low. So most species have got less than 10% overall lipid composition. So they're low fat, which is good, right? But as you remember, most of them are packing the punches in these good fats. Okay, so if we look at these 4 Australian species, the Cystophora tullulosa, which as you might recall is the one I said was looking best for our seaweed salad, and one of the other seaweed salad species that we tested, and then the red alga and the green that we haven't yet tested with our taste test. These guys are also, so these are the ones with the highest lipid concentrations. They've also got the highest concentrations of omega-3 or high concentrations of omega-3 and omega-6. Whereas the nori, which had overall very high percentage of omega-3 fatty acids, has got a relatively lower proportion of fats overall. So the total amount of lipids and the proportion of those individual fatty acids is important when you're thinking about the overall consumption. And from a fibres perspective, pretty much all the species are looking pretty good from the perspective of crude fibre. It was highest in the hijiki, and the other species are all looking at around, again, in the 5 to 15% range. So, and our Australian species faring quite highly in the crude fibre. Now, crude fibre does differ to dietary fibre, and we haven't yet been able to measure dietary fibre, which is the important thing to be able to measure. So with these preliminary analysis, we can get an assumption that with these values that would translate to dietary fibre, but they're not necessarily directly translatable. So what does this all mean? Well, essentially what we think it means is that these Australian species that we've looked at are looking pretty good from both a taste perspective and a nutritional perspective. So they're performing well on these tests, these preliminary tests that we've done. I haven't at all spoken to you today about the differences in time that we found, but we did find temporal differences in nutritional values. So when you harvest the species is likely to be important for when, the nutritional value, and also potentially impact on the taste associated with those species. So there's still a lot of work to be done looking at when is the best time to harvest our Australian species to both optimise taste as well as that nutritional punch. So there's a lot of work yet to be done. This is only the, fledgling stages of what is, potentially a very much bigger project before we can see these seaweeds getting to market. We are interested in doing additional nutritional analyses, and at the moment we're in the process of trying to get some extra funding to do some of this work. We're particularly interested in the first place in looking at sterols, which phytosterols have been shown to reduce cardiovascular disease and reduce the bad cholesterol in the bloodstream and therefore assist with cardiovascular health, diabetic health, and a whole range of other issues. And also, one of the things we're interested in and concerned about is looking at the mineral composition and the metal contamination. So, as I said, we need to collect seaweeds from clean waters, and seaweeds are good sources of these microelements and that are depauperate in our terrestrial soils, so they're potentially really good sources of minerals for us, but some of these minerals can become toxic in high concentrations. So we need to have a good understanding of the natural levels of various minerals in the Australian species that we're looking at, and indeed in any species of seaweed that we're eating. And we need to know where they're coming from, from, and how much variability you're getting in those mineral concentrations and potentially toxic metals from areas around urban areas, for example. So I think it's important to understand the natural levels, but also the potentially affected levels by urbanisation and industrial or domestic runoff into the ocean, so that we can make informed choices about where to best harvest these species or where to best farm these species on a commercial scale. We've only tested 6 of our species with our taste trial. We've got nutritional data for 9. There's a plethora of more species out there that are potentially good to eat. So this is our first step of the first suite of species. There's lots more species that we can look at and lots more dishes that we can potentially try those with. And of course, importantly, before this can go to market, we need to understand the impact of harvesting these species. So if we're going to look at wild harvest, we need to understand the ecological impact of any harvesting procedure on both the species that we're collecting itself, but the flow-on effects that that might have on any of the invertebrates, fish and other seaweeds that live amongst those seaweeds. Many of these species are forming important habitat, and it's quite likely that if we take out large amounts of it, it's going to have an impact. So, but there are harvesting techniques that can be used, and just as we don't, you know, the clear fell of a forest is much more detrimental than selective harvesting. We can apply similar processes in the sea with potentially minimal to no impact. But understanding how those interactions work needs to involve more ecological research. And to be able to send these species to commercial aquaculture, we need to understand more about the basic biology. For many of these species that we looked at, we have very little biological information about them. And so to take it to a commercial aquaculture stage, we need to understand lots about the life cycle and the growth conditions and the optimisation to be able to propagate these plants and get them growing on with maximum light conditions and nutrient conditions and all these sorts of things. So there's a lot more research that we need to do, but we think it's looking pretty promising, and that sometime, hopefully in the not too distant future, we may start to see some of these Australian species which with potentially harvested in much cleaner waters than our Asian counterparts gracing our plates. So to finish, I'd like to firstly put out a big thank you to all our funders. We couldn't have done this work without the support of the public and we couldn't have done the work without the support of our wonderful volunteers. So thank you all to those that have come today, and to those of you that weren't able to come, thank you as well. Masaru Natsuno was our chef who created the dishes, and we thank you very much. There's been lots of media promotion of this work, and that was integral for getting the funding in the first place, and indeed the other teams that were involved in the research on collaboration with Possible were instrumental. And with that, I would like to open up to take any questions from the audience across any of these councils. Thank you very much.
Yeah, thanks very much for letting me sit in on that. That was really interesting.
Sure.
I had a question about, so are you going to, do you think you'll try the Japanese method with, is it sea stock that there's a texture that was sort of letting it down?
Yeah, I think it would be interesting to look at further and certainly to get more details about that processing method. We obviously did research processing methods before we began, but that we didn't find that information. So I'll be talking more about that with my Japanese colleagues and trying to sort of really nut out that processing technique. And I'm hoping that maybe next year I might get myself over to Japan and actually visit some of these processing pans, and then we can actually, get some better first-hand information about how the processing is done and try and replicate that better. And of course, which is in a way fantastic, because now we have this baseline nutritional data for the more crude processing that we've done. If we then do the more diligent processing, if you like, that can potentially improve texture. We've got the ability to then compare that and see how that's affecting the nutritional value. But one of the other things that I didn't mention is that the hijiki is actually being shown to be high in inorganic and organic arsenic. Now, in the quantities that people eat, it's considered not to be a large problem, but it is a potential health concern for countries that are importing it into their areas. So hijiki has been banned from importing to America and some European countries, and Australia is looking at it for restricting imports as well based on the arsenic levels. Now, personally, I think it's potentially a little overreactive because we need to look at not only just the total arsenic, but also the arsenic speciation. But we're really interested to look at our Australian alternatives to hijiki, if you like, to see how they're faring, particularly in regards to arsenic speciation.
Sounds good.
Okay. Any running Warrnambool with questions?
How many people were in the trials?
I'm not sure if you guys can hear the questions here, so I'll repeat them. The question was how many people were in the trials, and yes, I probably should have mentioned that. We had, I think, 45 people in total, so we ran 5 repeat trials with up to 10 people in each trial. So, and we had a range of demographics there. So, range of different age groups, range of nationality backgrounds, and but the only criteria that we selected was that they needed to have some history of eating seaweed. But for most people, that was that experience was consuming nori with the sushi. But we did have, I think, out of our 45 people, I think we had 15 people that were either Japanese or Chinese heritage living in Australia. So a much longer association with eating seaweed and more diverse types of seaweeds. And
do you plan to harvest from the sea or opportunistically from the shore after storms? Harvest by sea?
The question there for those at the other ends was, do we plan to harvest from the sea or opportunistically the stuff that's washed up on the shore? And the answer to that, I think, needs to be based for food. We need to be looking at fresh harvest from the sea because of the potential for contamination for stuff that's washed up on the shore. And we never quite know how fresh it is. There is a big industry, or on Australian standards, a strong industry based around Deakin, the bull kelp, which is washed up on the shores of King Island, and that's harvested commercially. But they harvest the storm wrack and then they send it off overseas, and it's used for extraction of alginates, which is one of the polysaccharides components in the cell walls, and it's got great commercial value for a range of different products, as an additive to a range of different products. But I think when you're dealing with food, we have to deal with food safety standards as well, in terms of both collection as well as in preparation of the dishes. So certainly all the collections that we've made have been fresh harvest. But as I said, we don't really understand the implications of that harvesting, and so we harvest it in a way that, as an ecologist, I have, I guess, an assumption of how we can best harvest that to reduce impact. So some species, for example, have got rapid growth rates and can revegetate or regrow fronds if they're broken off. And other species, if you break off, so some species you can take the whole plant and just leave the base and it will grow back. Other species, if you take it and leave the base, nothing's going to happen. That's the end of the plant. So understanding those growth cycles and growth forms of the species can help us to harvest more sustainably. One of the species that we tested for our work, Holmesara maxii, which is an intertidal species, we know from all the ecological work that we've done in Australia and New Zealand, where it also occurs, is that it's incredibly important for biodiversity across our shores. So when we're harvesting that, rather than just clearing a whole patch, we know from our ecological work that if we take a frond from here and a frond from there, we can collect a relatively large amount of biomass without having an impact on the shore. But where we clear, areas of the shore, that can have a big impact. So at the end of the day, we might end up with the same amount of seaweed in our basket, but the method that we use to collect it can have a detrimental or not effect on the environment and the associated species. So for Holmesara, we have that an understanding of those thresholds of what's possible to clear and what's not. But for most of the species, we get to understand those ecological relationships. Yes.
How is seaweed harvested in Japan?
The question was how seaweed harvested in Japan, and the answer to that is it depends on the species. So for most of that commercial 10,000 tonnes that's harvested commercially is from mariculture products. So in most cases that involves culturing the algae to a given stage in the lab and then seeding it out on some sort of structure and to then grow out to complete its life cycle in the ocean. So, and that is typically done on ropes or baskets or nets or some sort of structure depending on the species and how it best grows. And it might be horizontal rafts or vertical nets, again depending on the species. And then it's simply a matter of just coming along on the boat, lifting up the nets and stripping it all off the rope. So it's quite quick and quite economical. Harvesting in the ocean does also occur, but it's not always done well. So in California, for example, they've been harvesting the giant kelp, which we get on our shores here, and it's an incredibly important habitat for a range of fish and commercially important species as well. What they did but it grows incredibly quickly. So in North America, they've been harvesting it for the alginate, so there's products in the cell walls that I was talking about with the bull kelp as well. But it grows at a phenomenally quick rate. So they basically go over the ocean and in a boat and mow the lawn effectively. So they're chopping off the tops and then it will regrow. And then they've got such massive expanses of forests in California of this particular species that they can basically harvest areas and then go back, on the next round and then regrow. So for in some parts of Europe, they're also harvesting ascophyllum, which is a fucoid alga that's related to the Holmesara that I was talking about before. And so it's an intertidal species, but it's much longer. So the Holmesara might get to lengths of say 30 to maybe 50 centimetres at the most. The ascophyllum is grown to lengths of over a metre. So it's a much more structurally complex habitat. And again, we know from some of the ecological work that's been done that it's incredibly important habitat. And thinning of that canopy can reduce the ability of that species to shade the organisms underneath that are associated with it. So if you reduce the canopy to a given level, it's effectively the same as having no canopy. So, but typically the way they harvest that is by raking. So they go over the shore and rake it. So they're not being cognizant of the fact that this is important habitat. They're just clearing big patches. And again, that species is actually very slow to regenerate. So when I was in France last year, I saw the impacts of this harvesting on a relatively small scale, but you can just look at the shore and you can tell that they've been through harvesting. It looks like they've gone through a big little haircut. And you can see the impacts of that on the rest of the species. And I would just hate for that sort of thing to happen in Australia. I think we've got a really unique opportunity to do it right from the the outset. So it's a potentially fantastic new industry that we can bring to Australia, but I would strongly caution that if we're going to do it, let's do it carefully. Let's not rush in hammer and tongs. Let's make sure we fund the research that we need to know before we jump in at the deep end, and then say, oh, sorry, we messed up.
There's concern about the generational practice of training the horses on the beaches and the risk of tetanus or infestation.
Yep.
Because we're immunised against tetanus, we don't have an incident, but if it's imported, exported to areas where there's not immunisation, it could be lethal.
So, I'm not sure how well you can hear the questions at the other end. The question was around, in Warrnambool we have horses that are trained on the beaches, and so the question was around the potential for tetanus infestation of seaweeds via the horses, and of course we have immunisation here against tetanus, but if we're exporting the seaweeds to other parts of the world, they may not be resistant. Is that, do I interpret your question correctly? I think that risk is relatively low because we are collecting our seaweeds away from, we've been careful to collect our seaweeds away from areas that might be likely to be a problem. But I think the message that you're sending is that, it is a really important one, because I think we do need to be very careful about where we collect these species from, how we collect them, and to make sure that we're both not messing up the environment, but also potentially putting people at risk. So from various health perspectives, and some of that is the potential for disease transmission. The other is potential for toxic compounds of heavy metals, pesticides, those sorts of things as well. So I'm not familiar with any research that suggests that tetanus could be transferred by consumption of seaweeds, but that doesn't mean it's not possible. It's a bit outside my area of expertise.
Spreading as fertiliser.
Fertiliser is probably less likely to be a problem because it is the trial processes that go on in during fertilisation is you get composting and heat and that's associated with that. So the heat is likely to impact on those viruses as well and reduce that disease risk greatly, I think. Of course, seaweeds have been shown for a long time and across the world to be great fertilisers. And because they are this fantastic source of trace elements that are missing in our terrestrial soils. So, particularly in Australia, a bit more seaweed compost would be, and seaweed tea would be a great thing for our veggie gardens.
Would spores survive composting?
I don't know that, and that would be something that should be looked at. But again, if you're not collecting the material from areas where there's that potential risk, then you should be out of harm's way. So certainly if that was a concern, then you'd be advised to not be collecting your seaweed from areas where the horses were being trained, for example. Okay, I think unless there's any more questions from Tassie, I think we've exhausted our questions here from Warrnambool. All good in Tassie?
Thank you.
Okay.
Yeah, thanks. Very much.
Thank you very much for taking the time to come today, and again apologies for the technical problems that we had in the beginning, but hopefully we did get there at the end, and hopefully you enjoyed the presentation. Thanks again.
Congratulations, Alecia.